So my research aims to target a protein that's upregulated in pancreatic cancer and see if blocking this protein can enhance chemotherapy. I've shown that when I block an intracellular part of the protein, cancer cells become more sensitive to chemotherapy. So this suggests that this intracellular part may be a potential therapeutic target. I plan to look at other intracellular parts of this protein and then try to target them through a peptide-based approach.
The core goals of my research are to evaluate whether diverse pastures, including multi-species forage, can improve animal performance and support more sustainable pasture-based sheep production systems. So to date, we have found that multi-species forage have improved yoke condition and lamb performance, including lambs reaching their slaughter weights earlier using less nitrogen inputs. So this has shown real potential for productivity, economic and environmental sustainability gains. Going forward, I'll continue to assess animal performance and greenhouse gas emissions, including how change in the botanical composition of animal diets influences these responses.
The core goal of my research is to develop and microbially validate novel processed meat products to replace nitrite and therefore reduce nitrosamine formation. The key results I have to date is the development of 3 promising nitrite alternatives that have been tested in 3 different processed meat products: bacon, ham and salami. These have been tested against Salmonella typhimurium and a surrogate for Clostridium botulinum. I have also explored the natural spoilage organisms of the 3 different products under different packaging systems. My future work involves looking at the microbiota of the 3 different meat products with the 3 separate alternatives using 16S sequencing.
So my research looks at how we can improve vitamin D intakes and status in Ireland, especially in groups that are considered more at risk, like breastfeeding women, older adults and children. And we're hoping to do this using a staple food like bread. So what we're seeing is that low vitamin D intakes and status are still a very real issue. We found that fortified foods already have a very meaningful contribution to intake, so this supports the food fortification approach. We ran a randomised controlled trial in older adults, and we saw that fortified bread can significantly increase vitamin D levels in this population over the winter time. So we've just wrapped up another RCT in families, and that's our final one. So hopefully with the results of this we can explore whether or not the intervention worked differently across different age groups, and this can hopefully add to the evidence base that supports food fortification as an effective public health strategy in improving the population's vitamin D status.
So the aim of my research is to investigate the Irish underage national field hockey pathway athletes, their lived experiences and demands, and we're looking into their injuries, performance and wellbeing metrics. So far we're in season 2 of data collection, and we found that 74% of all athletes reported at least one pain or injury throughout the season. And out of these injuries, 76% of them were time loss. So that's some of our preliminary data so far. So we're currently in season 2 of data collection at the minute, where we're collecting weekly surveys. And we're also planning our qualitative paper, which interviews participants across the pathway. And then finally, we have a performance testing session in UCD planned, which evaluates a testing battery of power, strength and speed.
My research focuses on early recognition and management of community-onset sepsis in Ireland, particularly in general practice. So far, we have found high mortality among people aged 45 years or above due to sepsis, and that clinical outcomes are influenced by high chronic disease burden among Irish population. Going forward, we are collaborating with emergency departments in Ireland assessing community-acquired infections. We are also assessing the burden of sepsis among people who had a history of sepsis in Ireland. We are also generating evidence on post-sepsis care models for improving outcomes among this population.
The core goal of my research is to shed light on the molecular basis of atrial mechanotransduction. That is the proteins that underpin how a physical stimulus is detected and transduced into an electrochemical signal. The key results to date are the automated mapping of the atrial receptive field in rodent models and the demonstration that there is an amiloride sensitive channel that mediates atrial mechanotransduction. Future work will focus on confirming the presence of ENaC in the atrial tissue and determining if it plays a role in the cardiac cycle.
The goal of my research is to see if a non-pharmacological intervention will help sleep in people with advanced cancer. So we're using cognitive behavioural therapy for insomnia, bright white light therapy and an activity plan. The key results I have is that it is acceptable, it is feasible, and the non-pharmacological intervention has helped with sleep, it's helped with symptoms, and it's also helped with quality of life. Well, my study to date was a feasibility study, so the hope is that we will secure some funding in order to do a much larger randomised control trial in order to provide more definitive results and inform clinical practice.
Well, my core goal of my research is try to understand why the same kind of mutations develop into a severe disease in some cases and in some other diseases doesn't create any kind of disease, and how PALOX plays a really important role in all this. So our key results is that we have seen that PALOX are enriched among those genes that has been seen as lost in healthy individuals. But more importantly, we have seen that not all PALOX contribute equally to this to happen, and the way they arise is really important to understand this role. So the next logical question that we have in our research is about understanding why some PALOX can tolerate the loss of one of their genes and why some others, once they have been seen as lost, they develop into a disease. So to do that, we are going to include some other features related with protein complex and also expression tissues to understand how do they behave.